Good afternoon, everybody. Welcome back.
Recording in progress.
Good afternoon, everybody. How you doing today?
Good.
Good, good. I think we've got 15, 19, and 20 to do today. I hope you've had a chance to fill in your notes. I'm going to be going pretty quickly through them, so we have time at the end that I didn't have before. But again, at about 3:30, regardless of our activities, I do need to go pick up my kid. So hopefully we can get through everything we need to by then. Okay, does anybody have any questions before we get started with today's PowerPoints?
Will you be going over the exam, the last exam?
Yes, for anybody who has it, but we're also going to be prepping for this week's exam. So I'm going to try to move quickly through the notes so I'll have time to go over the review questions and then also go over the last exam with people who are still here.
Thank you.
Okay, you're welcome. So let's go. Chapter 15 is what I am starting with. So hopefully people can get caught up. If you come in late, you can always rewatch this for it. Okay, autonomic nervous system. This is basically reflexes. Okay, so what is the sequence of events in a reflex? There's the stimulus, something that happens to you that causes the sensory receptor to be stimulated. Then the signal goes from the sensory or afferent neuron towards the CNS. There's CNS integration. That integration would be in the spinal cord unless it was a cranial reflex, something happens in your head. Then immediately an efferent or motor neuron is stimulated. Then that goes to a target tissue. It's almost always a muscle or a gland, by the way. And the response is movement. Usually the response can also be a glandular secretion, as we saw in insulin. And then there's going to be feedback to the CNS, letting you know whether or not the response has been adequate to take you back to homeostasis. Okay, there was no figure, so don't worry about it. All right, so what are some classifications of pathways? Well, one is called the effector division. This means something that makes something happen. It's the motor division. So one division of the effector division is called the somatic, and that goes to skeletal muscles only. So this would be conscious. Autonomic would go to visceral or digestive organs. This would be almost always smooth muscle, could be glands. Integration site, another way to classify reflexes is, are they spinal? That involves reflex below the head, or is it a cranial reflex? It says brain, but the kind of reflex would be cranial. So what are neurons in the pathway? If it's monosynaptic, it's 2 neurons. Now, that might seem a little counterintuitive. Mono means 1, but how many fingers do I need to have a gap between my fingers? Right? So if you're going to have a gap between your fingers, you have to have 2 fingers. So monosynaptic is 2 neurons with 1 gap. Polysynaptic is 3 or more neurons with 2 or more synapses. Okay, so this is not a figure on your test, but it does show you the difference. This is a monosynaptic reflex because you have the receptor, the sensory neuron. Please note on the sensory side, the cell body is going to be in a ganglion outside the spinal cord. And then on the efferent neuron is going to have its cell body inside the gray matter of the spinal cord. So it goes directly from the sensory neuron right out to a skeletal muscle. The difference here is in a polysynaptic reflex, you're going to have the same kind of thing, but inside the spinal cord in the gray matter, you're going to have an inner neuron. What inner neurons do is they make a decision. Do I want to pass this along? Do I want to repress it? Do I need to involve any other body parts? You're going to see that in upcoming reflex. And then the same basic idea though. Okay, so visceral reflex is a little different. It almost always affects smooth muscle. It's always polysynaptic, so that's a little different. What are the functions? It can cause goosebumps to form. Now, why do goosebumps form? Well, in our distant ancestors, they had fur. We have hair, a little bit of hair across our body. So for example, on your arm, you can get, if you get creeped out or cold, the hairs will stand up. So why does that happen? Well, it turns out that the hairs are at an angle and there's a muscle behind it. And when it pulls, it lifts the hair up and the skin with it. So when you're lifting a hair up, the skin bulges upward as the hair lifts. That's the goosebumps. And that's a reaction either to being creeped out or to being cold. Now for us, it doesn't make us any warmer, but for our ancestors who had fur or a lot more hair, it worked well. And you can see that in cats and birds where they fluff up when they're cold. Okay, you can regulate internal organs, especially those involved in digestion, and help maintain blood pressure. This is how blood is directed to different parts of the body depending on the source of need. They integrate either in the spinal cord rather than part of the brain, but officially it is part of the brain. Okay, skeletal muscle proprioceptors. One is called a muscle spindle. You find it in muscles, as you would guess. I'm going to go back and restart that. You find a muscle spindle in muscles, as you would guess, that senses the degree of stretch, so how much the muscle is stretched out, so you know how much it is. Golgi tendon organ is a little bit different. That is near tendons, as the name would imply. The key here is how much force is on the muscle. So you can regulate how much force the muscle is generating by this sensor. Joint receptors sense the pressure on the joint and the angle of the joint. So there's 3 different basic types of proprioceptors associated with skeletal muscle. Okay, a myotactic unit. So what is a myotactic unit? Shown here in this picture. You have a receptor, sensory neuron, goes through the spinal cord, just like we saw, a monosynaptic reflex, and then the motor neuron. The idea here is if you're holding a book or a brick or whatever, and more load is added, the reflex is to contract more unless it overweighs you, and you can't hold it, then you drop it. So there's 2 different kinds of reflexes I'll be going over. So a myotactic unit is all the pathways controlling a single joint. Example, elbow joint, all the nerves, receptors, and muscles act together as a myotactic unit. If you look at the name of it, myo means muscle, tactic means tactile or sense of touch. So a combination of muscle and touch. What happens when someone adds weight to the load you're carrying? So if it's a weight that you can easily handle and someone adds an extra book, you just contract your muscle a little bit more without even thinking about it. It happens as a reflex. Now, if somebody puts a whole extra ton of books on or something really, really heavy and you're already carrying a large weight, you will drop it as one of your reflexes. Okay, so response to excessive force. So force pulls collagen fibers, which squeeze the sensors. So that would be one of the Golgi tendon organs. The muscle increases recruitment. That means more of those motor units are recruited. But if you try to pull hard enough and you're still not maintaining the load, then the reverse happens and inhibition of contraction happens and you drop them. So it just depends on whether you can hold it or whether it's too much for you to hold. All right, the knee jerk. You guys have heard that expression, knee jerk reaction. Now they use that in language because it means that somebody reacts automatically without thinking about it. And let me get into the chat here. Okay, someone's just saying hello. Tendon strike with a rubber mallet stretches quadriceps group, causing a reflexive contraction. You guys have seen that. Someone puts their knee up on a table and then you strike just below the patella with something rubbery or whatever, and the knee goes out. Now, one of the activities that you're supposed to be doing this week is to simulate that. Now, you want to find something similar to a rubber mallet. Now, if you don't know what a rubber mallet looks like or how big they are, look it up on Google. But you want to strike just below the knee. And if you're doing it to somebody else, well, you can't do it to yourself, by the way, because you anticipate it. But you need to do it to somebody else or have someone do it to you. So someone should strike just below the patella with a bent knee and you'll see the foot kick out a little bit. Now, why does this happen? Well, what it is it's a reflex that keeps you from falling down. You may have experienced this. For example, you're at Disneyland, standing up all day, your quadriceps group is tired. And all of a sudden you feel your knee buckle. But do you land on the ground? No, you don't. You feel your legs very quickly straighten out and you pop right back up. That is the functional use of this quadriceps tendon strike. And the idea is that the muscle is lengthening very quickly, but you didn't intend on it lengthening. So the idea is you're going to fall if your knee bends too much. So the signal is sent to the quadriceps group and you straighten your leg and then you don't fall. The antagonistic or hamstring muscle is inhibited because the idea is you want to make sure that you straighten your legs so you don't fall. How is this reflex useful in walking, running, etc.? Well, we use this reflex to down on a bent knee when we run. So if our knee is bent and we feel their legs starting to bend even more, the reflex is to straighten the legs so that we don't come all the way down and fall while we're walking or while we're running. Okay, so this is the reflex. I don't know if this one is on your test, but we will see at the end of today if we can do that before. Similar in size and weight. By the way, do you think you would get a similar reflex if someone hit you with a sledgehammer as hard as they could in the knee? Would you get that reflex?
No, it'd probably break your knee.
It would break your knee. So if you see that on the test, are you going to pick that?
No.
Hitting with a sledgehammer. Okay, I'm giving you a big hint. One of the wrong answers is hitting someone in the knee with a sledgehammer. A rubber mallet and a sledgehammer are nothing alike. So don't pick the one that says sledgehammer. Okay, I was saying don't pick the one that offers sledgehammer. Okay, so when you tap this, what happens is by deflecting this patellar ligament inward or patellar tendon, depending on how you define it, the muscle gets longer really quickly, but not by very far. What that does is it sends a signal to your spinal cord, and it does two things. The first says, don't contract the hamstring. We don't want to lock our knee. We want to straighten our leg. And the other says, contract the quadriceps group. So what it's doing is the simulation, your body isn't smart enough to know this is a mallet. It thinks that you're falling. And so by straightening the leg a little bit, it keeps you from falling down. So this is the efferent motor. And then the dash line, by the way, whenever you see dash lines in these figures, it means inhibition. So you're going to inhibit the antagonistic group, which is your hamstring, and you're going to flex the quadriceps muscle. And the action is it swinging out forward. So you're going to need to simulate that as part of one of your labs this week and tell me how it worked and how effective it is. All right, flexion reflex. You guys have ever put your hand on something really hot?
Yes.
Do you leave your hand on something that's really, really, really hot and is burning you?
No.
No, you don't. So what do? You pull your hand away, and often you pull your hand away even before you know that there is anything happening. By the time you realize it's hot, your hand is already starting to move away. So there's a pain stimulus, the nociceptors, which are receptors that generate pain, generate action potentials. There's spinal integration, often stimulating more than one motor nerve. For example, all the muscles on the back of your shoulder and the back of your arm would be trying to get you to pull away. The flexing the appendage away. And this is a very common reflex because what we do is we put our hands out and our appendages out into the environment. And then when there's danger, we pull it back towards us. So the flexion reflex is the most common because it's safer close to you than away from you. So extension moves your appendages away from you. Flexion pulls them closer to you where it's safer. And then you feel the pain. So the interesting thing is we convince ourselves that we're feeling it before we move, but in reality, your arm starts to move at exactly the same moment your brain receives it, and it rearranges the order to convince you that you felt the pain before you moved your hand, but your hand is actually moving at the exact same instant you feel the pain. Okay, so this is a different kind of pain reflex. This is a more complicated one. Now, this one is not going to be on the test, but it does show you that you can have two different pathways, several different pathways. One on the extensor inhibited. So normally you would be stepping down, but because you stepped on something sharp like a tack, you switch this, you lift this foot, and then you bend this knee and kind of hop forward. You guys have all done that, especially when stepping on Legos, which are by far the most painful thing to step on. And I'm not sure why that is. I think Lego engineered it that way. So anyway, this isn't on the test, so I'm gonna go ahead and bypass this. Okay, so there is some information about this figure that could end up as a review question. You will need to know it. The signal is sent to the opposite leg. Extensors are stimulated. Flexors are inhibited. This is the opposite of what you would normally be doing walking. And the body is supported so you don't fall down. All right, movement, coordination of several muscle groups. A reflexive movement requires spinal integration. So it has to go to the spinal cord, and then a signal comes out. Input goes to the brain. Now, postural reflexes are those that involve skeletal muscles, but generally are not conscious. They're controlled through your pons or your cerebellum, which are semi-voluntary. So there's cerebellar integration. That's the cerebellum, which is the brain below the brain. This is almost always subconscious motor movements. It helps you maintain balance. And then you get input to the cerebral cortex, which is conscious. So you get information about what's happening. The actual movements, you don't. And so, for example, if you're a piano player and you've been playing for a long time, you don't have to think about where your fingers go anymore. They go there automatically. That comes from the cerebellum. The only thing you have to think about is if you're a seasoned piano player is how fast do I want to go? Do I make it a little faster, a little bit slower, a little bit more accent here or there? But you don't have to think about moving your fingers anymore because the cerebellum, which is that little brain sitting underneath your cerebrum, integrates all that if you've practiced. All right, feed forward, anticipates body movement, reflexive adjustment to balance change, prepares the body for threat, blink, duck, or tuck and roll, and combines with feedback to let you know what's happening. There's not going to be a lot of questions on this, so I'm just sending it out there. Okay, visceral movement. Now, moves products in hollow organs. In this case, it would be cardiac smooth muscle. It can act as a valve. That would be a sphincter. In the digestive tract or blood vessels, you're going to have circular rings of muscles called sphincters. There can be multiple controls, autonomic neurons and hormonal. If you remember me telling you that smooth muscle can be stimulated by hormones, not just through motor neurons. Okay, so that was it for 15. That went pretty quick. And so let's go ahead and go to 19 and 20, the first part of 19 and 20. Does anybody, by the way, have any questions about what we just covered?
No.
Yeah, it's pretty straightforward, and I hope that you guys had a chance to fill in your notes beforehand. Okay, 19 and 20, cardiovascular physiology. The way this is set up, because it's heart and blood vessels, I'm combining both chapters together. So if you are trying to find this information in the text, you'll have to search between those two chapters. Okay, so this is a picture directly from your OER text showing you the pathway of blood. Now, I may be asking you on one of the tests to tell me to trace the pathway of blood. So generally where we do is we start in the right atrium. So I'm going to go through it quickly. So if you don't take notes here, rewatch it and take detailed notes. Because I may ask you to trace the pathway of blood, including blood vessels, valves, and functions of all of the structures that it goes through. So I'm going to go through that quickly. Here we go. We're in the right atrium. It's going to receive deoxygenated blood, shown by this kind of purpley blue color, from the superior vena cava and the inferior vena cava that's coming in at low pressure. That pulls up here. The right atrium contracts, pushes through the tricuspid valve. The way to remember that is the word right has R in it, so does tricuspid. They both have the letter R. So it pushes through the tricuspid valve into the right ventricle. Now notice the right ventricle is on the left. Why is that the case? Because you're facing the person and it's their right and their left, not your right and your left. So when you're facing someone, your right is their left, your left is their right. So that's why right is on the left side of this picture. So it goes into the right ventricle. Then when the ventricle starts to contract, the tricuspid valve closes. This pulmonary semilunar valve opens, the blood pushes up into the pulmonary trunk, which is here, and then the pulmonary arteries. Normally, we would associate arteries with red blood, oxygenated, but if it has the word pulmonary in it, it's going to be reversed because pulmonary, going to your lungs, is doing the opposite job of all the other arteries. Most arteries move oxygenated blood to the tissues and veins move deoxygenated back. But in the pulmonary system, the reverse is true. You're taking deoxygenated blood to the lungs and bringing it back oxygenated. So when you see the word pulmonary, the colors and oxygenation levels are going to be reversed. So the blood goes out to the lungs and then comes back through the pulmonary veins, which are red because pulmonary means reversed. It goes in right here to the, let's see, we're on the right side. So this would be the left atrium. It opens the mitral valve or bicuspid valve. Another name for the mitral valve is the bicuspid valve. Blood gets pushed by the atrium into the ventricle, which then stretches out. When the ventricle contracts, it pushes up and out past the, are they showing it here? Yeah, the aortic semilunar valve. So that opens and allows blood to go up through the aorta and up through the body's blood vessels. Then when it goes to the body's tissues, as you can see in this image down here, it comes back up to the heart through the inferior vena cava and down to the heart from the head and shoulders to the superior vena cava, and you have completed the circuit. So if I ask you this question, you trace the pathway of blood, including all blood vessels, major blood vessels, and the functions of each. So be ready for that on the test. I believe that's next week. Okay, the heart is a 4-chambered structure. Not all vertebrates have that. Fish have 2 chambers. Most lizards have 3. You have the right atrium and ventricle, which beat together, by the way. It's not left side, then right side. It's 2 atria contract and then 2 ventricles contract. The pulmonary circuit is to the lungs and back. You send out deoxygenated blood, which is indicated by the blue color, back to the heart as red. Now, please note, blood is never blue. It's only coded that way. If you've ever had blood drawn at the doctor's, you've noticed that it's a dark brick red color. That's coming directly out of your veins. And if it came out blue, that would be an idea that your venous blood is blue. It's not. It's brick red, and then it goes to bright red when it gets oxygenated. But they code it blue to help you clarify whether it's deoxygenated blood or oxygenated blood. So then the left atrium and ventricle is going to be receiving blood from the lungs and then sending it out to the body through the systemic circuit and then back to the heart deoxygenated. Blood vessels are what are called closed circulation. That means, with one small exception, blood always stays in blood vessels. Now, that's barring injury. The only time barring injury that blood is not in blood vessels is when a woman is pregnant. She has blood circulating around the placenta outside of blood vessels, and I'll show you that when we get to our very last chapter at the end of this class. Arteries are always blood moving away from the heart. Now notice, arteries and away doesn't mean all arteries contain oxygenated blood. That's true on the systemic side, but reversed on the pulmonary side. Capillaries is the only place naturally anything ever enters or exits your body. Now, when I say naturally, I mean outside of a clinical or medical environment. You can inject something directly into a vein, but outside of some medical intervention, the only time anything ever leaves your blood or enters your blood is through a capillary. Veins always move blood toward the heart, normally deoxygenated, shown in blue, but in the pulmonary circuit, it would be red, oxygenated. Okay, what are some pressure changes that happen? Blood always flows down a pressure gradient from high to low. It's the highest at the heart, which creates the driving pressure for blood pressure, and it decreases over distance. And we'll see that it doesn't decrease in a linear way, it decreases in almost a logarithmic way, and I'll explain to you why that's the case. Hydrostatic or hydraulic pressures in the vessel, that's the pressure of the fluid trying to push outward against the vessels. And it decreases 90% from aorta to vena cava. And you'll see in some of these figures, it actually shows it dropping all the way down to 0. So that would be 100% drop. And I'll explain why that happens when we get there. Okay, so this is a figure that I'm pretty sure is going to be on your exam, and I'm going to be asking you to tell me about it in detail. So I'm going to go through it in detail pretty quickly. My strongest recommendation is you rewatch this after I post this video. Pause it, take notes, start it, pause it. You guys get the idea, so you can get all the key information down. It's very clear to me when I'm grading your essays who's done this and who hasn't. If you do it and you take lots of notes about what I say, you'll have a really good essay to put together. Another quick reminder is you've got to start all your essays with a summary sentence, which tells me what the whole figure is showing with a pretty good amount of detail. So if you just say this figure shows how pressure changes in the blood vessels, that's kind of boring. And it doesn't suggest a 5 out of 5. It suggests more like a 4. Let's go through it. Along the y-axis is millimeters of mercury. This is the pressure in various parts. And along the right are the various structures that they would be in. So we're going to start at the aorta, which is pumping oxygenated blood out of the left side of the heart. Now notice we're going to be talking later on about what is mean arterial pressure. But in this essay, you are going to want to talk about mean arterial pressure, or MAP, what it is and what it tells you. That's coming up in the notes afterwards. So you want to include that in this. Okay, so notice that we have ventricular contraction. They're showing a little thing like this, but more or less it's just a straight drop in pressure. So when it drops down from, let's say, 110 to 70, that's ventricular diastole, which means diastolic pressure, which then means the ventricle is relaxing. The only thing they don't show here is what is happening in the ventricles. In the ventricles, the pressure during diastole would go all the way to 0. What they're showing in this figure is what is happening just in the blood vessels. Now, the question is, why doesn't the blood pressure drop all the way to 0 during ventricular diastole? It's because the blood is trapped inside of a vessel. And as it stretches out, there's back pressure put on it by the elastic fibers in the aorta and other large elastic arteries that prevents it from going back to 0. So basically, imagine putting water into a water balloon. And then if you let off on the top, the elasticity of the balloon will push the water out. And that's exactly what is happening. When you force blood into the aorta, it's going to stretch it out. It's going to store energy in the elastic fibers of the aortic wall. And then during ventricular diastole, the pressure doesn't drop to 0 because you're pushing the blood down the line during ventricular diastole. Okay, so you see it's going up and down in the elastic arteries, even a slightly greater variation than in the aorta. But what you'll see is once we get past the elastic arteries into the muscular arteries, we see a big drop off. The biggest drop off that we're going to see is in the arterioles. Look at how fast the pressure drops from the beginning of the arterioles up at around 100 to the end of the arterioles down to 20. That's almost 80% of the total drop happens in the arterioles. Why is that the case? A couple of things. First is there's a great amount of bifurcation. Cross-sectional area, total volume available. So as the blood, what was the last thing you guys heard me say?
Great amount of bifurcation.
Bifurcation. So that means lots of splitting into different arterioles, which are generally not named. And what it also means is there's a greater cross-sectional area, which means the blood can kind of expand out in the way that a river slows down during when it goes into a wider channel. So it drops off quickly because there's so many more arterioles than there are muscular arteries. And so that's going to represent a huge drop. Now, later on, we're going to compare what is happening to velocity when we compare it to this, but that's coming up in a later thing. So what we're going to see is mean arterial pressure is calculated in a way that I will explain to you in an upcoming slide, which you need to talk about. So the pressure is going to start to drop. It drops off the most quickly in the arterioles for the reason I told you. Then in the capillaries, we're going to see again a drop, not as great. Notice it's shown as purple. Purple indicates that you're going from oxygenated to deoxygenated blood. So you're giving up oxygen and taking in CO2. And by the time you make it to the venules, now you're fully deoxygenated. The blood pressure doesn't drop off very much. And then at the last minute in the vena cava, at the end of the vena cava, just before you get back to the right side of the heart, the atrium, it drops off basically to zero. Okay, there's lots more information about this in your OER text, so check it out. And there's going to be more information in some upcoming slides. So give me lots and lots of detail and make sure you talk about what mean arterial pressure is and how to calculate it in your answer to get 5 points. Okay, now we're going to be talking about fluid flow here, and these figures are not in your text, but I do want you to know them. Now the question is flow rate. Now if we're going to talk about flow, in this
Is this
situation,
not what you were wanting us to include in our answer for the last slide that you showed?
yeah, mean arterial pressure, what it is and how you calculate it.
Which is the calculations that were just on the following
Yeah, it's
slide?
going to be on some upcoming slides. What is
Got
MAP,
it.
mean arterial pressure? So just make sure you get it in there. And if for some reason I don't cover it, which I'm pretty sure I will, you still need to look it up and tell me what it is. So instead of just saying, oh, mean arterial pressure is the gray line, you need to tell me what it is, and I'm almost 100% certain it's coming up in one of these slides coming up. Okay, so flow rate, liters per minute. Now, if we have an incoming pressure of 100 millimeters mercury and an outgoing of 75, and a different vessel, we have 40 and 15, which one has the greater flow? Would it be the top one or the bottom one, guys? Which one would have the greater flow, the 100 to 75 or the 40 to 15?
The 100 to 75,
Why?
because it's larger diameter? I'm not sure.
Well, is there anything on the screen that would tell you which one has the greater flow?
No.
There's
This-
nothing in the picture that tells you which one has greater flow.
No,
Give your
besides
head
there
right
being
here.
the number 100.
Well, both of them are 25 at the wait. Yeah.
Yeah.
The
I'm
flow
trying
is
to trick
equal.
you guys a little bit to see if you're paying attention.
I had
The
to
difference
look
between
like
100
twice.
and 75 is 25. The difference between 40 and 15 is 25. And it says
Oh.
right there, flow is equal.
So
That
they are
means-
equal.
They are equal. It says it right there. And I was trying to see if you would look and say, well, Mr. Sage, they're equal because it's the difference in pressure, not the overall pressure. So now you get it. Okay, flow velocity. And we're going to be talking about velocity. It's different than pressure, and it has a slightly different dynamic. And I'll explain what the dynamic is when we get to the slide that shows all the different things compared to each other. So the velocity, how fast the blood is moving, velocity is equal to the rate, how much blood is moving divided by the cross-sectional area of the vessel. Now, how is it possible that the Mississippi River is moving thousands of gallons per second of water if the water is only going past you very slowly? I want you guys to think about this. How can the Mississippi River down by the Delta be moving thousands of gallons per second if the water's not moving very quickly?
That's a trick question.
It is kind of a trick question, but you got to look right here.
Mhm.
If the velocity is low, what has to be high? You guys remember numerator and denominator? Okay, you're not in it right now, but that slowly is because it's so wide. Does that make sense? The Mississippi River is really wide, so the water slows down even though you're moving a ton of water past that point because it is so wide. I'm so angry at my internet. I almost bought what is called a I can't remember. It's a hotspot thing that you can take cellular data. I think I got to do that because this
I'll pitch in.
this internet unstable thing is so freaking irritating. Okay, so the reason why you can move so much water through the Mississippi, even though it's moving slowly, is because it's really wide. Now, the question I have for you is, could you move the same amount of water through half as wide of a river?
That would not work the same way.
Same amount of water past a point if it's half as wide. And
It would not flow the same at the same rate, no.
Right, it go twice as fast. Does that make sense? So the reason why the Mississippi slows down is because the banks are so wide. If we made the banks artificially narrow, they would go faster. That's what they're saying. The velocity or speed is equal to how much is moving divided by the cross-sectional area. And what we saw was that the cross-sectional area of the arterials was great, which is going to make the pressure drop, but it's also going to make the velocity drop in an upcoming slide. Okay, so one of the key things is resistance slows flow. And if you want to see how this works, try the following. If you have two different, do you guys know what a boba straw is? Everybody knows what boba is, right?
Yeah.
Okay, boba straws are twice as big as regular straws, right?
Mhm.
So if you want to see how this works, try breathing through a boba straw and try breathing through a regular straw. Which one do you think will be easier?
The bobo straw,
Why?
because it's wider.
Because it's got a bigger diameter, right? So what we're going to see is the vessel diameter, both in liquids and in air. The smaller the diameter, the more the flow is going to decrease because that increases resistance. That means frictional force against the wall. Now, why is that the case? A very large vessel has more center and less edge. A small vessel has more edge compared to the center. So a small vessel is going to have more resistance due to frictional forces between the fluid or air and the walls. I hope that makes sense. The other thing that's going to slow you down is blood viscosity, how thick it is. Now, thicker gives you decreased flow, but what would be a benefit of having thicker blood?
You would bleed less.
You would what?
You'd bleed less.
Possibly, but I don't think so. It has to do with kind of injury.
Well,
Blood
wouldn't this have to
viscosity-
do kind of with like an INR of the blood? So the thicker the blood is, the less likely you are to have an issue of extreme bleeding. Whereas if you were to have a thinner blood, you would have the potential of
It's
bleeding.
possible, yes, it would leak out less. That's not the main reason why we have different thicknesses of blood. Now, thicker blood can be due to dehydration, but the main reason why people have thicker blood is because they've exercised more. So what happens is a hormone is sent to the red bone marrow, telling the red bone marrow to make more red cells, and more red cells get packed into your blood, making it thicker. Imagine adding more chunks to a fluid. It makes it thicker. That means you can carry more oxygen, but then the blood is thicker and harder to pump, which means your heart has hydration, not due to lack of water, but because their blood becomes so thick, their heart can't pump it anymore. That's often the source of death due to dehydration. So much to tell you, so little time. Okay, now, tube length. Longer tubes cause decreased flow. People who are really obese have a lot longer blood vessels because they have to get out into their adipose tissue. Now, if you want to simulate this thing, do this. Take a regular straw and try breathing through it. Then cut that in half and try breathing through it again. Which one do you think will be easier to breathe through? The long straw or the short straw?
Short straw.
Short straw because there's less resistance, less wall to pass along as the air gets inside. So the longer the tube, the more resistance there is, and the less flow there is generally. Okay. I wish I had more time to talk to you guys about it. There's actually a really cool simulation called PhysioX that I couldn't assign for this semester because it's not visually impaired. I can't even remember what it's called for people who can't see. It's not helpful for them, but it has a really good lab on that I used to use. Anyway, moving on. Let's talk about the heart, a little bit of anatomy of the heart. The pericardium is a membranous sac surrounding the heart and protects the heart from rubbing against bones and other tissues near it. The chambers are both left and right, and atria and ventricles. That's what this stands for. Coronary vessels means any vessel supplying the heart. You can supply the heart with nutrients and oxygen.
Today's the day.
You find valves.
Stingray migraine.
There's two different types of valves. There's the atrioventricular valves, which have these large flaps, chordae and papillary muscles, and then there are the pulmonary semilunar valves. Excuse me, both semilunar valves, both aortic and pulmonary. The difference is the atrioventricular valves prevent blood from going from the ventricle back to the atrium, so they have to be highly reinforced, whereas the semilunar valves only prevent blood from going from the aorta and pulmonary trunk back into the ventricle, so they need less reinforcement. And last but not least, the myocardium is the muscle tissue of the heart. It's called the myocardium. So this kind of repeats all the same pathways that I showed you earlier in the lecture. Okay, cardiac muscle cells. I think I talked about it a little bit when we were talking about muscles in general. Autorhythmic means it can contract without, oh, that's a mistake. Oops. Guys, please note, it should say can contract without stimulation from the body. So.
I was wondering what that was. I was like, that doesn't sound right.
So it cannot contract with stimulation from the body?
And can contract without stimulation. Okay, it can contract without stimulation from the body. I left out the word without, because with and without are opposites, right? So the idea is once you get a cardiac muscle cell contracting, it'll do it all by itself without any further stimulation. That's what autorhythmic means. So anybody who's not watching the lecture is going to get it completely backwards. And sorry for those people who don't watch. Okay, so let's look at the intercalated disc. This is where one cardiac cell touches another. They contain desmosomes, which are cell-to-cell connections which transfer force. That's the physical glue between the cardiac cells. Gap junctions, they pass electrical signals from one cardiac cell to the next so that you can start a signal anywhere in the heart and the entire heart will contract together as a single unit. That's called a syncytium. You may need to know that. Many mitochondria. Do you guys remember what mitochondria are for? When we were talking in the very first chapters about cellular respiration, do you guys remember what mitochondria create, what they generate? What do mitochondria do?
Create ATP.
ATP, right. So question, can cardiac cells easily go? Yeah, someone says, if you have noise in the background, please mute yourself. It might be Dakota. I'm gonna go ahead and mute you. Remember, unmute yourself if you want to say something, but otherwise keep yourself muted. The idea is that the cardiac muscle cells can't undergo anaerobic respiration because what happens after a certain amount of, let's say, running, you get to stop and rest. Does your heart get a chance to rest for a while if it goes into anaerobic respiration? What do you guys think? Can the heart take a few minutes to rest if it goes into anaerobic respiration?
No.
No, it's
No,
beating
because what
faster.
happens if your heart rests for more than a few seconds?
Throw a clot.
You die, right? So the longest break your heart muscles ever get from the first day of your life to the day you die is 1 second. 1 second is the longest rest your heart muscles ever get. So what that means is they have to stay constantly in aerobic respiration, regardless of how hard you push them. That's why there's so many mitochondria. That's why there's so many blood vessels, and that's why there's so much glycogen stored in this muscle tissue. Large T tubules. You also want to make sure that the signal spreads all the way through this relatively thick cell so that all of the sarcomeres can contract together, and they have a similar sarcomere orientation that we saw in skeletal muscle, but instead of just one really long thin cell, it's going to be a single nucleus and much shorter cell, but it's kind of branched, so it's a little bit different, doing a very specialized job of pumping blood. Okay, now I may be asking you to tell me about what's going on during an ECG or EKG. So what this is showing, and what I'm going to ask you to do on this, which is what I don't normally do, I'm going to ask you to go to the book and learn a little bit more with what is going on during ECG. And I almost never do that, but I'm assigning it specifically right now. So what it's showing is not all the details of what's happening during an ECG, and it's in your book, but it's not in this PowerPoint. So during atrial diastole, which is relaxation, we have this flat area here. Then when we go from diastole resting to atrial systole or contracting, get the P wave, which is a little bit of electrical signal. Notice that it's much smaller than the QRS complex because the atria are much thinner walled. They don't have to do very much of a job other than pushing blood down into the ventricles. Now, why do they have to push blood into the ventricles? Because, sorry, getting ahead of myself. If the ventricles don't stretch out enough, they're not going to be able to contract with enough force. So what atria do is they push the blood forcefully down, forcefully stretching out the ventricles so that they can snap closed, pushing the blood out with a much greater force. But given that, it's going to be a smaller bump because there's so much less myocardial tissue in the atria than there is in the ventricles. Okay, now during ventricular diastole, this means atrial systole means the contraction of the atria. Ventricular diastole is the resting of the ventricles, but right here where the ventricular systole, and it should be scooted over just a little bit to the left, contraction of the ventricles. The beginning of ventricular systole actually begins right at Q.
Professor, you froze.
Yeah, what was the last thing you heard me say?
Ventricular systole.
Okay, ventricular systole, this section here is only showing the physical contraction of the ventricles. Electrical ventricular systole begins at Q. So this is you guys remember how an axial potential works? It's kind of like a summation of axial potentials happening through the ventricular myocardium, going from Q to R. And as soon as it gets to R, then the ventricle starts to contract, and that's why they showed it as ventricular systole. But the beginning of electrical ventricular systole goes from Q to R. Rest, and then right after the ventricles get done contracting, we have the T wave, which is repolarization of the ventricles, which actually causes a little bump. This is the ventricles. So now we have ventricular diastole. So for some part of the cardiac cycle, both chambers are going to be at rest, but notice that you never have both ventricles contracting at the same time. One of the things that's a little bit misleading about this figure is there's a small gap between the end of ventricular systole and the beginning of ventricular systole so that the blood can be pumped from the atria down into the ventricles. It takes half a second or a quarter of a second, so it's not instantaneous. So they should really have scooted this atrial systole back to Q, left a little bit of a gap right where the E is, and then started ventricular systole just after Q. But you guys get the idea. You guys need to look this up. You need to tell me as much as you can about what an ECG is, but I got to move on. All right. This is what I was talking about. Characteristics of cardiac muscle cells. There's a strength-length relationship. Now, if you remember me talking about the length-tension relationship in skeletal muscles, and that was that if there's a fairly good amount of overlap, then you get the most power. If there's too much overlap, you get very little power. And if there's very little overlap in the sarcomeres, you get very little power. Now, it's different in cardiac muscle. In cardiac muscle, the more the heart is stretched out, the more power it has to contract. Now, this leads right to a thing called ventricular hypertrophy or an enlarged heart. Have you guys heard of enlarged heart or ventricular hypertrophy?
Yes.
Okay, that's a problem. Now, normally you would think bigger heart is better, right? Bigger beats harder. It's not true. The reason why ventricular hypertrophy or enlarged heart is bad is because it means that the heart pumps less forcefully. Now, why is that the case? Only a certain amount of blood is going to go in there. Now, if you have an enlarged heart, what's going to happen is the blood's going to go in there and it doesn't stretch because the heart's already mostly open. And if it only stretches out a little bit more, it's not going to be able to beat very forcefully. But if you start with a smaller gap and you stretch it way out, that gives you much more force to beat. So that's what the problem with an enlarged heart is those cardiac muscle cells are not being stretched out, not able to generate adequate force during ventricular systole, which is ejection force of the blood, or stroke volume. Stroke volume is how much blood leaves the ventricles per beat, and stroke volume drops with ventricular hypertrophy or an enlarged heart. Lots of stuff to tell you.
Question.
It has what is called a long action potential. Now, if you remember, action potentials in a neuron are pretty quick, and they come bleep, 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 bleep really fast. But in a cardiac muscle cell, the action potential is much longer. And what that does is it prevents summation. I don't know if you guys remember me talking about what summation or TREP is. That is, if you add more calcium, you get more and more and more contraction up to a complete fused tetanus. So I'm squeezing my hand as tight as I can. Question I have is, would you ever want the heart to contract and hold for a second? Is there ever any reason you want to have it contract and stay contracted?
No.
No, it has to relax instantly. And you don't want summation where it contracts and goes into tetanus, because you would die. So what happens is, during the contraction, there's a mechanism built into the cardiac muscle cells that has to do with calcium and a whole other variety of things I'm not going to ask you to know, so that if you stimulate the heart during ventricular systole, it's not going to, there's no summation possible, no TREP, no fused tetanus like we would see in skeletal muscle, because in skeletal muscle, you would want to squeeze and hold to hold on to something. But in the heart, never the case. So this long action potential and large refractory period. Dextrocardia, I'm not as familiar with. Goodness, I'm gonna have to look that up. That's not right on the tip of my tongue what dextrocardia is. If you can define it for me, I could probably explain it, but it's not on the tip of my tongue right now. So if I see you chime in with what it is, I can talk about it, but right now I don't have anything to say about dextrocardia. Okay, so this long refractory period means you cannot have summation, you can't have increased force, no fused tetanus or any tetanus at all. There's always going to be a
Professor?
contraction and a complete relaxation.
Dextrocardia is when the heart's on the opposite side of the heart or the chest rather than in its normal positioning. So it's shifted over.
It shouldn't affect the heart's function. I don't know if there are other issues associated with its impeding in the lungs, but normally if the mediastinum, which holds the heart, has enough room, whether it's pointing down into the left or down into the right, shouldn't make a big difference whether or not it functions adequately. The biggest issue with the heart, as far as I can tell, is faulty valves, leaky valves, or what do we call that? It's called a murmur. Heart murmurs can cause issues, or what is that? Fibrillation, atrial fibrillation. I mean, there's a whole lot of heart issues. But I was saying there's a whole lot of heart issues, but I think whether it's on the other side of the okay, so sympathetic and parasympathetic pathways, this is directly from your text as well. We see at normal rest that we have, these are the contractions, the membrane potentials here for the heart. When we have parasympathetic stimulation, that's what happens when you sleep or are resting. Notice that there's greater distance between them, and so your heart rate's going to slow. In sympathetic stimulation, this is the fight or flight, or basically any exercise. We have faster depolarization, the heart rate speeds up. That's more or less what this is trying to show. Sympathetic nervous system speeds the heart rate, and the parasympathetic slows it. So this is what it's showing it. Not a whole lot of details here that I need you to know about. AP stands for action potentials. They're generated from the autorhythmic cells in the sinoatrial node, here on out abbreviated SA or SA node. It spreads down through the internodal pathways, and that internodal means between the SA and AV node. The pathways, that's generally in the posterior wall of the atria and across the atrial myocardial cells. Okay, so they're saying it's two separate things, but it's basically the same thing. And then when that happens, the atria start to contract. There's a pause. And this is one of the things that they didn't show you in the ECG, is that there's a slight delay between the end of the atrial contraction or systole and the beginning of ventricular diastole. And the question is why? Why does there need to be a pause? Well, it takes a moment for the atria to finish squeezing and for the blood to transfer through the AV valves into the ventricles. And so if the ventricles started to contract right after the atria, they wouldn't have a chance to fully fill up with blood. They wouldn't be able to eject with enough force and enough blood leaving the heart to do its job. Then the AV node to the bundles of His, bundle branches and Purkinje fibers. This is all the going down into the interventricular septum, the septum between the left and right ventricles. And then right and left ventricular contractions happen from the apex upwards. Now this sounds a little bit confusing. Doesn't the apex mean the top? Like the top of a house is the apex. Well, in anatomy and in physiology, well, really anatomy, the apex is almost always pointed downward, the opposite of what you would think. So for example, the pointed part of the heart, you know, the point on a cartoon heart shape, the pointed part is the apex. Also your nose, the apex is here, down at the bottom of your nose. So what we're going to see in various parts of the body is the apex is pointing downward, not upward. So why does the contraction start from the apex and go upward? Well, the question is, down at the bottom.
Can you repeat that? You froze as soon as you said apex.
Okay, so did you guys hear me talk about how the apex is pointed downward in anatomy rather than upward?
Yes.
Okay, so why is it that the contraction of the ventricles starts at the apex, which is the bottom, and moves its way up? Well, to understand why, I gave you the analogy of where do you squeeze a tube of toothpaste? Do you squeeze it up where the hole is or down at the bottom? And why? What do you think?
Because where the blood exits the heart is at the top.
Right? But I was talking about toothpaste. Where do you squeeze the tube? Up by the exit or down at the bottom?
By the exit? I think it depends.
Okay, how do you think
It drives
most
me nuts if
people
you grab
squeeze
it
the
from
bottom.
the exit.
squeeze their toothpaste? Do they start at the bottom or they squeeze it right on the top?
The bottom.
The bottom, because it's coming out the top, right? You want it, because if you put your hand over the top of the toothpaste and squeeze, it would get all over your hand, right? And it would
You
also
also
want
end
to
up
push
with a
the
bunch of
toothpaste
leftover-
down and out.
You end up with residual stuff at the bottom too. That
Right.
Pushed up.
Right. So we start at the bottom, which is the apex, and we start contracting upward because all blood leaves the heart up and out. Remember, the aorta is coming up and out, basically this way, and the pulmonary trunk is going this way. Pulmonary trunk goes across the front. The aorta goes behind that, out the other way to the left. So we start at the apex because we want the blood to go up and out. We don't want it to start up at the top because then it would be pushing blood down towards the apex, be less efficient. That's what I wanted to get you guys to understand. Starts at the apex. That's why the signal spreads through the Purkinje fibers down through the atrioventricular septum and starts down there at the pointed part of the heart, moving upward because the blood has to be pushed upward. And as it starts to squeeze, we want to start at the bottom, pushing blood up and out, just like toothpaste. That's all I got to say. Here we go. Now, check it out. This should help you with the essay on the this EKG, ECG. So what it's showing you is what is happening in the heart associated with each of these parts of the EKG. So what you're going to want to do is use this figure to help you explain what is going on during the different parts of the EKG. Now, I may put this figure on the test, or I may just put the figure that I already showed you on the test, but either way, you need to explain what is happening in each of these sections by looking here, okay? So it shows you what is going on during this part of the EKG. So remember I said QRS is the beginning of ventricular systole. So here we have the ventricle stretched out, beginning of the QRS, then we have ventricular contraction, pushes the blood up and out. Then during ventricular relaxation, we have the T wave, which is repolarization. And then we have all of the chambers filling with blood. And then the P wave is atrial contraction, pushing the blood down into the ventricles. Then in this little gap here, the little gap between P and QR is where the atria are pushing blood down into the ventricles. Remember, you do need to get into the book. Give me a lot more details from the book. Please don't copy word for word anything you see out of the book. If I suspect it, I'll open it up. And if you've copied word for word, you lose all the points for that figure. Okay? So what are some information that you can gain from the ECG. Now, back in the day, they used to use paper EKGs. Does anybody still get EKGs on paper?
No.
I kind of
It's
like
on a
them.
machine, but it prints out.
It'll print it out. But back in the day, there used to be like these little needles with a little pen on it, and it would actually go bounce up and down, and it would actually be like a strip. Now what they do is they record it in the printer. I mean, they record it in the computer, and then the printer prints it out so you can look at it. But back in the day, it used to record it, in real time. Okay, it can tell you signal conduction. Are the pathways in good shape? All of those internodal pathways, Purkinje fibers, bundle branches, etc. We can tell you heart tissue status. Is there damage? Has a person had a heart attack or a myocardial infarction or any cardiac event recent? It'll tell you that. It'll also give you conditions or pathologies like heart murmur or anything where the heart hasn't been built correctly, some congenital issue. Okay, so what are some steps in the heart cycle? Well, remember I told you I may be asking you what is going to be happening at each of these steps. And here it is. So we're at late diastole now. All chambers relax, filling up with blood. But blood is just basically pouring into both the atria and ventricles. Now, please note, there's blood going from the atria into the ventricles even before atrial contraction or systole, because everything's open and the blood's pouring in from the inferior and superior vena cava, deoxygenated blood, as well as blood from the lungs. We're going on both the left and the right. So all chambers relax, filling with blood. But atrial systole, what it does is it adds 20% more. It doesn't seem like very much, but that 20% more is going to give that extra force that the ventricular myocardium or ventricular walls need to push the blood out with enough force to get it out to your body parts effectively to oxygenate your brain, your heart, and your lungs. Here's another interesting thing, a factoid. How much oxygen do you think the heart gets when the blood is in the chambers? Does the heart get any oxygen from the blood while the blood is in the chambers of the heart? You would guess yes, right? But no, the endocardium prevents any diffusion. So what happens is the blood has to come up and out of the aorta, and then it makes a quick U-turn and heads back down into the heart. So you have to have enough force pushing the blood up and out so that it'll go to the lungs. Even the lungs are living tissue, to the heart, which is living tissue, and then to the brain and the rest of the body. Okay, so this 20% more adds increased stroke volume and increased force of contraction. Isovolumic ventricular contraction. What does that mean? Both ventricles contract together, closes the AV valves, the louder of the two heart sounds, builds pressure. What happens is the atrioventricular valves are slamming shut as the pressure is pushing them closed. That's the very first sound you hear. In a similar way to a door in your house slamming shut due to the wind, that's what you're hearing. More steps. Ventricular ejection. It pushes open the semilunar valves, both aortic and pulmonary semilunar, and blood is forced out into those large elastic arteries. During ventricular relaxation, the aortic backflow slams the semilunar shut, which is going to be a quieter sound because they're smaller, less reinforced valves. So that's the dup. So it'd be lup dup, lup dup. So it's louder first because the aortic, the AV valves are closing, and then the second sound, the dup, is less loud because the semilunar valves are smaller and going to generate less force on the slamming shut. AV valves open, refilling starts, and we go back to the cycle. So this is the sequence of steps. So you're going to want to tell me about that. If I ask you, what are the cycle of steps that happen during ventricular diastole and ventricular systole? This is it. This is what you need to know. Okay, cardiac output. How do we calculate how much blood is leaving the heart? Well, it's usually around 5 liters. This varies between large people and small people, male and female. Males tend to have slightly higher cardiac output, females slightly less. If you're doing 72 beats per minute, that's not rest. That would be kind of walking around doing stuff around the heart, around the house, or walking slowly. You're going to get about 72 beats. I was just on the treadmill this morning, and right when I got started, I was at 72 beats per minute. So if you're getting approximately 70 milliliters per beat, you do the math on that, that gives you 5.040 milliliters or 5 liters. So you just move the decimal over 3 places and we have 5.04 liters. What is the heart rate? It's how many beats there are per minute. So if 72 beats per minute, it's going to be a little over a beat per second. The volume is milliliters per beat. So how do we calculate it? Well, we go from end diastolic volume, full, and in systolic volume, empty because it's contracted, and the residual is about 50%. This is surprising for many people because they assume during ventricular and even aortic systole that the heart is going to completely squeeze closed and then no more blood is left. If you remember the anatomy of the heart, there's all kinds of structures in there that prevent it from emptying completely. For example, the valves, the cords, the prevent the heart from completely emptying. So only about half the blood in your heart leaves on any single beat. Okay, folks, what was the last thing you heard me say?
Per beat, but you had skipped out just prior to that as well.
Yeah, I need to get one of those things. It's called a, I can't remember what it is, but it basically generates a Wi-Fi hotspot. I can't even remember what they call those. I need to get that for this because I'm running on my house internet. We're out in the middle of nowhere and we have crappy internet. Okay. About 50% of the blood leaves the heart in a single contraction. Okay. So now we are to review for this week's test, and then we're going to go over people's concerns or questions about the previous test. And we're at 2 o'clock, so I moved through this pretty quickly. So we're going to have plenty of time to do review for this week's exam and go over the old test for anybody who wants to. Please remember the way that the review of the old tests work is whoever wants to do it. Everybody but one person is in the waiting room as I go through the test with that person. When they are done, they'll leave. I'll bring someone else. It's like office hours. You only see one student at a time. So that's how it's going to go. Okay, so what do we have going for this week's test? It is chapters 10 and 14. So we covered that last week. I think what I'm going to do first of all, is I'm going to go ahead and go to the exam itself and show you the essays, just to make sure I get that done. So you know exactly what you need to prepare. Now, please note, folks, sometimes I don't check my email towards the end of the week. So if you want to run an essay by me, you want to get it to me as soon as you can, because I may not be checking my email as much as I should on Thursdays, Fridays, or Saturdays. So if you want to get it to me as quick as you can. And if I don't get it to you, I want you to know one thing, and that is that if you're really, really concerned whether or not your essay is top notch, it probably is, because I almost never get anybody sending me essays when their essay is terrible. Almost everybody who sends me an essay, it's good. And the reason why it's good is because you're the kind of person who would spend time pre-writing it and you'd be worried about whether or not it's good. And almost in my experience, I've been doing this 30 years now. Most people who are really worried about whether or not their essay is good have a really good one. So if you don't hear back from me and you put a lot of effort into your essay, then rest assured that it's probably going to get full credit on the test. Now, I'm not guaranteeing you that, but it's highly likely. Okay, so let me go back to here, and then I'll share it with you, the essays. Okay. Now, this is one of the things I didn't talk about. So what I'm going to be doing right now is lecturing on this figure. Okay. Should have done this last week. We ran out of time, but I'm going to do it this week so that you guys know exactly what I'm going to expect from this to get full credit. And so you want to, especially for this one in particular, you want to pause, take lots of notes, and then watch a little bit more and take some more notes and back up and rewatch it. Now, the reason why this is so significant is look at how many points there are. 10. That's double the normal points that you would get for an essay, because it's so complicated and because I'm asking you a complex 2-part question. And the second part of the question that's in bold, most people get wrong. I explain it, they instantly forget it, and they just go with what they already know. That will get you a poor score. But if you listen and take notes on what I'm saying, and then tell me about what I told you, you'll get a good score on this. So let's get started. So you don't have to take a whole lot of notes right now, but make sure you take detailed notes from the video recording. Okay, let's start with step one. And this is a good place to start. Sound waves are alternating areas of high pressure air and low pressure. That's what sound is, by the way. You guys have ever heard the riddle? If a tree falls in the forest and there's no one here to hear it, does it still make a sound? And most people say, well, it kind of depends on your point of view. No, it's very clear to a physiologist that the answer is no. Now, that doesn't mean only people could be there. If there was a mouse or a squirrel or a mountain lion, there would still be a sound. Now, the reason why, if there's no living organism besides plants there, it doesn't make a sound. And what I'm going to allude to you later on is that sound and pressure waves are not the same thing. What is sound? It's our ability to take those sound waves, convert it into action potentials, send it to our brain, let our brain decode it and interpret that as sound. Sound actually only exists in our brains. The only thing that exists out in the environment are pressure waves. So the answer to that question is no, there is no sound generated, only pressure waves if there's no one there to hear it, including other creatures, other animals. Okay, let's keep going through this. So in step one, pressure waves are coming with a particular frequency. Now notice in this case, we're talking about the frequency of the waves, how often these waves are arriving. That's one kind of frequency. Later on, we're going to see a different kind of frequency is the frequency of action potentials, and they are completely unrelated. So please pay really good attention that the frequency of the sound waves and the frequency of the action potentials have nothing to do with each other. So I'm going to get to that. So we have these pressure waves coming in, what you would call sound waves coming in. And when they hit the tympanum, which is your eardrum, it causes the tympanum to move back and forth like this. Now, the way that that works is, I'll give you an analogy. If you're standing on a dock in a lake or ocean or whatever, and a wave comes by, what happens to the dock when the wave hits it? It starts to move, doesn't it? The dock starts to move up and down, right? So as these pressure waves hit the tympanum, it starts making it move back and forth. Now, what is attached to the tympanum? Let me zoom in here. These ossicles, which are the malleus, incus, and stapes. So the malleus is right here, that's attached to the tympanum. The incus is attached between these two bones, and then the stapes is sometimes called the stirrup. Please always use the scientific term, is attached to what is called the oval window. So right here is where the oval window is. Now, because the tympanum is connected to these bones, the bones then start to move like this. As the tympanum is going in and out, the bones start moving back and forth. Now, this puts pressure on the oval window. Now, why do we need these bones? We need these bones for two reasons. What they do is they act as a series of levers. You guys ever seen that somebody could lift up a 200-pound boulder if they had a lever? That is a small rock and a long stick. You can put the rock near the boulder and pull down on the stick, and the big rock will move, even if it's heavier than you could move by yourself. It's because it's a lever system. Now, these levers, what they do is they decrease the amplitude, how far the movement is, and instead convert it into a smaller amplitude, greater force. Why do we need that greater force? Because inside the cochlea is liquid. So even though the tympanum is moving in air, all of the movements in the cochlea have to be in water. And so the lever system of these bones helps generate the extra force necessary to move the fluid in your cochlea. That's the first reason. The second reason is, even though you can't see it in this picture, there are muscles attached, the tensor tympani and stapedius muscle. What they do is when they contract, they pull on those bones and prevent them from moving. Now, why would you want to do that? Because the sound is too intense and you don't want to damage your cochlea. For example, there's a reflex that stops those bones from moving as soon as you start to yell. Have you ever noticed that when you yell, you don't hurt your own ears? You can't, because as soon as you start to yell, the muscles that are attached to these bones pull stopping the vibrations from getting in there. So even though the sound's really loud and it's right around the corner from your ear, it doesn't deafen you because you're anticipating. Now, if somebody else yells in your ears, it's going to sound really loud because it takes a moment for the reflex to kick in where you pull on those muscles and stop the vibration from coming through. A good analogy for that is when you put your hand on the top of a speaker or a drum, it can't vibrate as much and the sound diminishes. All right, you guys with me so far? Movement of the tympanum causes these to act as a lever system, putting pressure on the cochlea. Now we're in the cochlea, and here we are. So this spot right there, the oval window, the end of the stapes bone is right here. So now the stapes is pushing in and out as the tympanum moves, and it generates fluid waves. And as the fluid waves go through the scala vestibuli, they go all the way to the top, which is called the helicotrema. And then they come out the other side in the same way that if you push down on one side of a balloon, the other side is going to bulge up. If you push down another side, the other one's going to bulge up. My favorite visual example of how that works is, you guys ever seen someone sitting on a big balloon and a couple of people jump on it and throws them off into the lake? That's how in on the scala vestibuli, which is
We heard that's how, and then you froze.
Yeah, you froze.
yeah, I really got to get that little internet thing because my internet sucks. All right. So did you guys hear my analogy about when people are sitting on a big balloon and someone else jumps on and they fly off into the lake?
That was the last
Yes.
I heard.
Okay. So in this situation, the stapes bone is putting pressure in, which puts pressure on the fluid and pushes it all the way up to what is called the helicotrema, or top of the cochlea. And then the fluid waves come pushing out the other side, the same way that if you push down on one side of a balloon, another side goes up. So as you put pressure in, the bulge happens here. So this is why you have the round, this is where the round window is, and this is the liquid pushing out. Because liquid is incompressible, you can't make it smaller. If you push in on it, it has to be able to push out somewhere else. So as it's going in the oval window, it's coming out. So in and out, in and out. See like that? Because there's only so much volume in there. Now, as those waves are moving along, inward this way and outward this way, it causes this basilar membrane right here to start to vibrate. And that vibration strikes hairs that are in the organ of Corti, it's called. And that generates axe potential saying, hey, we're hearing a sound. You guys catch that? And the frequency here is going to be the same frequency as here. That's the frequency of vibrations in the liquid. Now, what's happening next? So now we have vibrations happening along this whole cochlea in the organ of Corti, the basilar membrane. So look at the question. It says, do not forget to discuss how volume and pitch are discerned. Now, I'm not asking you what is pitch. I'm not asking you to define pitch as the number of waves per second. I'm asking, how does your brain figure out pitch? How do you know whether it's a high pitch sound or a low pitch sound? And how do you determine if it's a loud volume or a quiet one? This what's in the bottom figure. The bottom one shows you how you figure out frequency. That is how you figure out whether it's a high pitch sound or a low pitch sound. And then I need to tell you how do you figure out volume. And you need to put this all together in a single essay. That's why it's worth 10 points if you can get this all together. And I will tell you, very short essays will absolutely not get full credit. Give me lots of details. Okay, so if we take the cochlea, which is normally shaped like a snail shell, and stretch it out so that it's flat, the apex is here, and then the area near the round window is here. So the way that this works is because this is thicker, this is thinner, if it's a high-pitched sound, like, it's really high-pitched sound, I can't go very high, because I got deep vocal cords. But if you hear a really high-pitched whine, it's going to cause the proximal part of the basilar membrane or organ of Corti or cochlea to move. Now, the way that your brain knows that you've heard a high-pitched sound is because signals are coming from this part of the cochlea and going to your brain. So it's completely linear. So it has nothing to do with the frequency tells your cochlea what part is going to move. So if this part is moving, that tells your brain, this is a high pitch sound. If this part is moving, that means it was a medium pitch sound, kind of like voice. And then if this part way out at the end is moving, it's a very deep voice, a very deep sound, right? So if it's a bass drum, it's going to cause this part to move. So it's completely location. Now, a better analogy for that is a piano keyboard. You guys know how piano keyboards work? On one side, you have low notes, and one side you have high notes, and in the middle, you have medium notes, right? Question, if I go over to where the low notes are normally, when you hit the key, it makes a low note. Is there a way that I could hit that key and make a high-pitched sound? Anybody play piano?
You can push your foot on the pedal and that will alter the sound a little bit, but it will not convert it to
No,
a
it won't.
higher
It
tone.
won't change it from a deep, like I go to one extreme, wherever the low pitch notes are. Is that the right or the left? I don't know the piano.
Left, I believe, is low pitch, right is higher pitch.
Okay, so in a piano, the direction is opposite from the picture. So if you go all the way to the left side of a piano and hit that last key, it's always going to make a low pitched note, right? And I want to say the pedals are whether or not you extend the note or make it shorter, but I don't play piano, so I don't know. But the point is, there's only two things that you can do with that key. You can hit it harder or less hard, and you can make the note last a little longer or less long. There's no way to make a high-pitched sound by hitting that far left key, right? And vice versa at the other end. So the way you determine whether you're going to make a high note or a low note on a piano is the location, right? Where on it you hit it. The only thing you can change is how loud the sound is and how long it lasts, but it's purely location. So in the cochlea, wherever the signals are coming from, that's how your brain knows that's what the pitch of the sound is. So it's purely location. You guys got that?
I have a question.
Sure.
What part of the ear is the one that when you get tinnitus due to a brain injury doesn't work anymore? And you are not able to hear it right, but when it's quietly, you hear this ringing sound?
Okay, so tinnitus is a ringing that can happen from one of two reasons. That can either be damage to the cochlea, or more often it's a spasm of one of these. Remember I told you there's muscles attached to these bones?
Mhm.
Sometimes what happens is the muscles go into spasm and they start, that looks really bad. They start pulling a little bit very quickly, almost like a very minor spasm. What happens then is it's sending a signal through the cochlea that there's a high-pitched sound coming through because there's that little bit of a vibration in the muscle. So that's one cause of tinnitus. The other could be damage to the hair cells themselves, which is misinterpreted in the brain as sound.
Can
Now,
that be, I'm sorry?
Go ahead.
Can that be fixed with surgery or hearing aids?
Sometimes what they can do is they can actually bypass the cochlea and put like those cochlear implants in. You guys ever seen the people who have the thing on the back of their head? What it does is it bypasses all this mechanical stuff and listens for the sound and sends it directly to your brain. And in that case, it would eliminate tinnitus because the problem is usually either in the organ of Corti along the cochlea or in the muscles attached to these bones. So if you completely bypass it, you would have to cut the cochlear nerve, which I don't know if they do that often, but that's one way to get rid of tinnitus. They can also do surgery to, I don't know the exact. Okay, guys, you want a little something special because you came to lecture today?
Mm-hmm.
Okay, do you guys know what the extra credit is normally on my tests? What do I almost always give you extra credit?
Lobsters?
Lobster. Would you rather do it on something more interesting?
Yes.
Okay, because you're here and because, well, you don't even have to be here. You can just be watching. If you want to answer a different extra credit question, not an extra credit, but if you want to substitute a Labster summary for what I'm about to give you, you can go ahead and put that in the extra credit blank instead of the question about Labster, and I'll give you as much credit as you deserve. And here's the extra credit question. What is tinnitus? What causes it? And what are the treatments? Write that down. If you want to answer that question and place the Labster extra credit, you can do that. And no, you can't answer both and get double extra credit. I know that's what you're thinking. And the answer is no. You have to pick either what's provided or what is tinnitus What is it? What are the causes and what are the treatments? And do not copy word for word off of Yahoo Answers or Wikipedia or any of that crap. If you want to look at lots of different things and get ideas, put it in your own words, that's great. But if I do an internet search on your extra credit and it comes up word for word, you get a 0 and you could potentially fail the entire test. So you guys know how to write essays, right? Tell me
Yes.
everything you can
Question
about
for
tinnitus.
you, kind of going
Yeah.
back to the tinnitus, with cutting the cochlear nerve, wouldn't that cause you to become deaf in that ear?
It would. It would. So then you'd be dependent on the cochlear implant. So that's what they do for people who have problems with these. If people have problems with their tympanum or the bones or the cochlea, and you can't hear for that reason, they just put a cochlear implant. So that would be one path, one way to get rid of tinnitus, but a pretty severe one, because it would mean then you're dependent on the cochlear implant to hear. And I don't know how often they do that. That is why I'm telling you, if you want to do some research on tinnitus and make that your extra credit question, that would be really cool. And I'd love to read all about it.
I have a question. My husband has tinnitus due to a brain injury. He served in Iraq and Afghanistan. So his ear loop is, he could, in order for him to hear me properly and understand what I'm saying, he has to read my lips. So when we went to the VA to see if we could get him treated for it, they said that we couldn't, just because they mentioned a name of a nerve that's attached to his head that also got damaged due to the impact.
Cochlear or vestibular nerve?
They said both of them. Like they're
the main nerve that goes out of there is called the vestibulocochlear nerve, and it goes to your brain. It sends signals about balance and about sound to your brain. So
yes.
my guess is what happened. I'm just guessing. There was an IED, right? And it went off, and it was unexpected, and it caused a lot of damage to his cochlea. And because the bones were damaged and the tympanum was damaged and the cochlea was damaged, he has tinnitus, right?
Yes.
Okay. Brain damage would generally be if something hit his head, if it was with a piece of shrapnel.
Yeah.
But if it was IED or an explosion went off nearby, that would generally be damage either to the cochlea, to the bones, or to the cochlea. I'm sorry, the tympanum, the bones, or the cochlea. So I'm guessing if it was a nearby explosion, it would have been that. And that often does cause tinnitus. My mom had it for decades because she went shooting with my stepdad without hearing protection. So anytime you have really intense sound that happens too quickly for that reflex that I told you about, that's why explosions are so dangerous for your hearing, is because they don't last very long. And by the time you respond to it by pulling on those muscles, the damage has been done.
No,
So,
his damage is due to shrapnel in his head.
oh,
So,
shrapnel in his head.
Yeah,
Okay,
he
but
landed on the IED.
but there could have also been damage to the cochlea because there was an explosive sound.
Yeah, because they said it caused him to have vertigo.
Vertigo,
So,
right. So
yeah.
that it could have been damage to his vestibulocochlear nerve, because vertigo has to do with your sense of balance, and that goes right through that same nerve. So that could have been damaged as well. I'm sorry, I got to cut you off, but I got to
No,
finish.
that's fine. I just wanted to know. Thank you.
I mean, I would love to hang out and chat with you, but everybody else is probably rolling their eyes and like, or
I
you guys all think it might be really cool, but I just want to make sure I have time to get through everything that we need to get through. So very cool, but time to move on. So the last thing I haven't talked about is volume. So remember, when I ask, how does your brain discern pitch? I don't want you to define what pitch is. I want you to tell me that it's what part of the cochlea is moving. Far away from the loud sound or a quiet sound of any particular tone. It has to do with how many exponentials are leaving. What was the last thing you heard me say?
Are leaving.
It has to do with the number of action potentials that are leaving the cochlea. Now, if you remember, I talked about the intensity in a nerve has to do with the number of action potentials per second. If there's a high intensity stimulus, there's going to be lots of action potentials going in a short amount of time. Less intensity, fewer action potentials going per time. Does that make sense? So volume is intensity. So the way that your brain codes for whether it's a loud sound or a quiet sound is how many action potentials are coming from a particular part of your cochlea. In addition, how many hair cells are stimulated. Here's the other thing. Some of the hair cells that stand up start to move when there's a quiet sound, and some require more vibration to start sending signals. So between the number of ax potentials coming from there and the number of hair cells sending signals, that tells your brain, was it loud or was it quiet? Okay, so loud, more hair cells are stimulated, more ax potentials come per second, which is frequency. See what I mean? The frequency of ax potentials tells you volume. It doesn't tell you pitch. Pitch is determined by where it moves here. So this is complicated and this is not easy. This is the most difficult essay I'm going to give you for the whole semester. That's why it's worth 10 points. So you guys think you can put that all together? Here's another thing I need you to do. For volume and pitch, how are they discerned? I need you to put that at the end, because notice that the question is at the end. So don't put it in the middle of your essay. Make it a separate paragraph. You guys got that? How volume and pitch are discerned has to be your last paragraph, or I may miss it. Okay? Make it a separate ending paragraph and you'll be good to go. Okay? That's the best I can do for now. Okay. This one is the sliding filament theory, and I did talk about this, how you have exocytosis of acetylcholine that opens sodium channels on the surface of the muscle cell that spreads through the T tubules. When the T tubule strikes the sarcoplasmic reticulum, that causes calcium to be released. Remember, this is the opposite of what's being shown here. Calcium is released, that causes the troponin and tropomyosin to move out of the ways, allowing the myosin head to stick to the actin, the G-actin, and then there's flexion. When you relax, then you get rid of these little circles. Calcium is reabsorbed. The troponin, tropomyosin then cover the actin, and the myosin can no longer attach. Now, even though it doesn't say it here, if you throw in how rigor mortis works, the idea that if you, wait, there it is. There's the rest of the figure. Now, this is what's happening right after calcium arrives. The tropomyosin and troponin moves out of the way, and then we have these steps. So I'm going to ask you not just to tell me what's happening at the top, but what's happening right there between the actin and myosin. And what is the troponin doing? What is the tropomyosin doing? And how does that relate to rigor mortis? So even though it's not prompting you specifically, what I talked to you about is the absence of ATP means that the myosin cannot let go of the actin, and that's why you get rigor mortis. So if you throw all that in together, you get your 5 points. I know it's a lot, but it gives you a lot to talk about. It gives you a much easier way to get enough detail to get full credit. Does that make sense? Now, I did talk about this, right? This is the patellar stretch reflex. Now, what is something that you could do at home that would allow you to make this essay a little bit more interesting?
Does that do the experiment with
the
the
lab.
mallet that you mentioned?
Right. It's one of the labs that you're supposed to do. So what if you threw the results of what you did at home into this essay?
Extra more better chance of getting full points.
You want your score to go up or down?
Up.
Up. If you want more points, tell me about what happened when you did it at your home. And that will show me that you're able to connect the laboratory portion and the lecture portion together. So instead of just talking about the steps in the figure, talk about, well, when I did it, or remember I even talked about when you're standing in line at Disneyland and you almost fall down, but you don't. All of those trying to write specifically what is this picture showing and nothing else, it's going to be harder to get a 5. Okay. What was the last thing you guys heard me say?
You broke up right after you said Disneyland.
Okay, so remember I gave you the analogy that when you're at Disneyland and your knee buckles and you don't fall because your leg straightens out. That was an example of how this reflex works. In addition, I talked about how when you're walking and the knee bends, you straighten your leg quickly so you don't fall down. In addition, you have a lab that you're going to be doing specifically about this. All those things that you throw in there make it a lot easier to get a 5 than if you just talk about the exact sequence of steps in this picture. I want you to elucidate, put in stuff from lab, put in examples that I gave, things like that. Okay?
Professor, isn't that supposed to be from the following week's exam? This question is the stuff that we covered today, which would mean that it's for next week's exam, correct?
That's
Or
chapter
are we throwing in both the two
No.
chapters that we covered today plus the 14 and 10 for this week's exam?
Well, wait a minute. Give me just a second, guys. Week 4. Lecture exam 10 and 14. I knew I should have left. Okay, so week 3. Huh. So week,
The same thing would be in correlation with the the ear question that you had. That would also be from the stuff that we covered today.
senses and reflexes are week 4. Yeah, if it's stuff, because look, you guys, we have already covered this. We covered this today, and you guys should have been able to get your notes filled in and work on the review questions and bring it to lecture and ask questions. And you don't even have to take this test until Friday or Saturday. That gives you 3 days to prepare from what. So just to let you know, the test is not always from what we covered last week. It could be partly from what we covered this week. But as long as we covered it, as long as the material was presented, then it's fair game for the exam. Okay?
We're taking exam 3 this week.
Right. 10 and 14 is what it says, but it should say 10, 14, and 15.
Okay.
Okay, I need to fix that. I don't know how that happened. Okay, let me fix that really quick. Sorry about that. Thank you for pointing that out. 10, 14, and 15. So thank you. So if you refresh the week 4 page, it will tell you that lecture exam 3 is 10, 14, and 15. So sorry about that, but we have covered it. So it goes up through 15. Okay, so yes, we already did cover it. So you guys have that information and you can get ready for the test. Let me do one other thing. Let me go back. Yeah, it was covered this week, but I just covered it. So you guys should be able to study between today and Friday when you're supposed to take the test, or Saturday. It should give you plenty of time to study. Okay.
Is that 4 essays in total? 4 short essays?
Let me share my screen.
Yes.
Okay, so what did we start with? We had hearing, right? Hearing is 1. Then we got sliding filament theory, how skeletal muscles contract is 2. Patellar stretch reflex is 3. So it's 3 essays and an extra credit. So a total of 4, but 3 that you need to do and 4 you can do if you want. Okay. So can you guys see what I'm seeing here?
Yes.
Okay.
Yes.
So you have 3 different options for extra credit. You can either tell me about the article, what's a PINA for? You can either tell me about a Labster lab, or you can tell me as much as you can about tinnitus, what it is, what causes it, and what are the treatments. So you have 3 options, but you can only do one of them. Okay, but at least you guys know now what the test is going to be on, and you have the material, so you should be able to prepare in the next 3 days for the exam. Okay, so now you know what the essays are going to be, and I do appreciate you reminding me that 15 is on there. So that was a typo. Which is, what did we say? It's 14. 10, 14,
10,
and 15.
14, and 15.
10, 14, and 15. And so what I would like you, there may be multiple choice questions from 15. So be prepared to see multiple choice questions on 15. Okay? So that
Could
was
we
a typo.
expect to see multiple choice questions for 15 on exam 4 as well?
There may be. There may be.
Okay.
Just have the notes ready to go and study them just in case. And remember, you guys have 2 hours to do this test. So if you see something from a previous chapter, it shouldn't be that hard to get your notes out and look it over quickly. But yeah, it would be a good idea to prepare for 15 in both this week and next week. Okay, so
You froze.
which is 10, 14, and 15. So please, if you guys did get a chance to fill it in and you did get a chance to work on review questions, that's what this time is for. So we're going to begin with chapter 10. If you guys, everybody, please get out chapter 10. I don't think I covered it in lecture because I ran out of time last week, but you still have the PowerPoint and you could have done your review questions using the PowerPoint and your notes. Who would like to start us off with a review question from chapter 10? Please remember to give the question number. I don't have the questions in front of me. Everybody else, and if you're here participating in this activity, please go to the question number the student, your classmate gives so that everybody's on the same page so that you don't have to say, what question number was that again? Okay, so please try to avoid that. So when somebody gives their question, you go there, everybody goes there, nobody has to say what question number was that. Okay, so John asks question 12, muscles can lengthen forcefully. They cannot. The only way muscles lengthen is if they're stretched out by an antagonist or by gravity pulling or some external force pulling the muscle longer. So by the constriction of or contraction of the antagonist on the opposite side. Okay, you're welcome, John. Anybody else either in chat or want to just chime in with a question from chapter 10? Casey says, number 5, when cells can contract without needing an action potential. I think you meant to say what cells can contract without needing an action potential from a nerve.
No, in the,
And
on the review, it says when.
Okay, so this is a typo. It should
asks-
either say what cells can contract or when cells contract without needing an action potential from a nerve, what is this called? So this one is probably not on the test because it has an issue, but I'm going to give you either answer. So if the typo was what cells can contract without needing an action potential from a nerve, the answer is cardiac muscle cells. If the question says when cells contract without needing an action potential from a nerve, what is this called? That is called autorhythmicity. So whichever way the test question goes, that's what it is. But I don't think it's on there because, as you've noticed, I give you lots more review questions than our test questions. And normally what I do is I remove review questions that have problems so I don't have to apologize for it or backpedal after the test. This one's probably not on there, but if it is, it's going to be in one of those 2 formats, and those were the 2 answers. Okay? Does that help, Casey? I know I'm going really quick, but we got to go. Anybody else for chapter 10?
number 18.
Okay, Hannah 18, and then John asked 32. So I'm going to go out to that after Hannah. Go for it, Hannah.
Okay, the more cross bridges that a contractile unit can form, the more what does it produce?
Did anybody get an answer for that? Tell me what you got.
I couldn't get it.
Okay. If you didn't get it, the answer is tension or force. Those are both correct answers. Now, I need to say something about my tests. If you expect 2 answers to be showing up and you only see 1, should you still pick it?
No.
Yes. If a right answer is provided, but you're expecting 2, that's still a right answer and you still need to pick it. I had one person say, well, I expected 2 right answers. I only saw 1, so I picked none of the above. Don't do that. The only time that 1 right answer isn't the right answer is if there's another one with it. But if there is 1 right answer and you know it's right and there's no 2 of these are right or all of these are right option, you got to pick the right one. So don't reject the right answer just because you're expecting 2 and don't see the second one. Does that make sense? The only time you reject the right answer is if there's another right answer and one of the choices is several or all of these are correct. I hope you guys can get that because somebody lost a point over that on the last test. Okay, John asks, number 32, why do skeletal muscles have multiple nuclei? Now, do you guys remember me talking about how skeletal muscles form embryologically from several separate cells and they all fuse together in a long cell? Now, why do they need to have multiple nuclei? Not only are they to guide protein synthesis, since there's so much protein in muscle, you have to have a near.
Professor, you froze when you first started answering that question.
Okay. Wonderful, excellent. Now, the reason why skeletal muscle have multiple nuclei is the following. They're fusion of multiple individual precursor cells in the embryo that turn into one long cell fused from several smaller ones, which means each one of them has its own nucleus. So that's the embryological reason. The functional reason is there has to be a nucleus nearby to guide protein synthesis. If you remember how DNA is read, and then you got messenger RNA and a ribosome and a protein bead, that has to be local. It can't happen on a microscopic scale miles down the road on the other end of the muscle cell. So by having multiple nuclei, it gives local protein synthesis to all the different parts of a very, very long, elongated cell. I know that's a long, detailed answer, but that's the complete one. Okay. Casey asks, number 20, the liquid internal portion of a muscle cell is called, did anybody get number 20?
Sarcoplasm?
Sarcoplasm. Remember, cytoplasm is the normal name, but when it's in a muscle, everything gets the sarco prefix instead of the cyto. Okay. So it's sarcoplasm. Okay. Anybody else for chapter 10?
It looks like someone was asking about question 5 a little bit ago.
I thought I handled 5. Yeah,
You got 5.
I did. I went through 5. And I
Number
remember,
23.
Can somebody read 23? Hey guys, if you put a question number, you got to put the question up there too.
I'll read it.
Because
I'll
I
read
don't
it.
have it. Hannah, read.
Which of the following is not something found in the sarcoplasm?
Okay, so what you need to know is go through the notes, find things that you would find in the sarcoplasm, and the right answer to that question is something that you would not find. Because what you're trying to find is something that doesn't belong with the others.
I'm going to read 22 as well. If muscles do their primary job, what 2 things do they generate?
What was the question number?
22.
22. Okay, did anybody get 22?
It says, what 2 things do they generate?
Okay, so one answer is either force or movement. That's one answer.
That's what
And
I
the other
have.
is heat.
The
Okay?
other is what?
So there's actually 3 things that they normally generate, which is force, movement, and heat.
Okay. Thank you.
Which may or may not cause movement and heat. So for example, if I just take my arm and I contract my bicep, did my arm move at all? Do you guys see my bicep popping up? I mean, I wish it popped up more, but did my arm move?
Yes,
No, my arm is still at the same angle. See the angle, 90 degrees?
but your
No,
arm.
I'm talking about
towards
movement in
you.
the joint. I'm talking about movement in the joint.
Okay,
So when I contract, there doesn't have to be movement in the joint, but there is a difference in tension. You guys catch that? And it generated a little bit of heat. So it's tension and or movement and heat. Okay, so force and heat, tension and heat, sometimes movement. If you don't have, the reason why my arm didn't bend is because I was also contracting the tricep, and the antagonist was also contracted. That causes an isotonic contraction. Isometric, excuse me, would cause isometric contraction. All right. John asks, number 17, what 2 structures are primarily moved by muscles? And what did anybody get that from the notes?
Got bones and visceral structures.
Bones and visceral structures. Very good. Okay, somebody else from 10. Question from 10. Anybody?
are we just covering 10A or are we doing 10A and B right now?
10A and B. We're doing both. I assume you guys were in 10A.
Yeah, we were.
Okay,
are.
let's go to 10B. Let's go to 10B.
Number 8,
Hey, read it.
the muscle cells of the heart are unaffected by outside stimulation. True or false?
Okay, question.
That's not 10B. Which chapter is that?
No, that's not the right chapter. You're right.
That's what I have on 10B as well.
10B.
Yeah, that's what I have.
Yeah, I
also have
Oh, is it?
That's 10B.
Okay. I think that's muscles, right? So
Yeah.
it's still talking about muscle.
Yeah,
So,
it's order. Sorry for the review question. So maybe that's why.
No, I think I just looked at the wrong number. Sorry.
It's okay. So is that in the correct chapter or not?
Yes.
Okay. What was the question again?
Okay. The muscle cells of the heart are unaffected by an outside stimulation. True or false?
Okay. So just because muscle cells can contract without stimulation, does that mean that stimulation does not affect?
No.
No. So even though they can contract without stimulation, they normally get stimulation from the SA and AV nodes. Does that make sense?
Yeah.
Or an external source. But just because they don't need stimulation doesn't mean that they cannot respond or normally do not respond. They normally are contracting by stimulation, but they on rare occasions can contract without. So that's false.
Okay.
That's a false statement. Okay, so we are in 10B. Anybody else from 10B? Chapter 10B. Anybody from 10B? 19?
21.
Hannah, can you read 19, please?
When you're trying to hold something in your arms for a long time and it is not very heavy, your muscle fibers take turns with the load. This phenomenon is called.
What do you guys have? Anybody have an answer for that one?
Asynchronous.
Asynchronous contraction. Remember, one set is pulling and while the rest are resting, then another set takes over while the other one is resting. That's called asynchronous contraction. Means not at the same time. Asynchronous contraction. Good. Casey, try to put the question in addition to the number if you can. If you
I
can
can
copy
read it. I don't mind.
Okay, great. Let's go more from chapter 10B. Anybody else? 10B.
Number 16.
16, read it.
When a very high intensity stimulus arrives at a muscle, which muscle fiber would contract?
Would it be low frequency, low threshold or high threshold?
High.
But the low ones would not?
Well, it says a high intensity stimulus.
Right, really
It'll be
strong
both.
stimulus.
Oh,
Both, because remember the low threshold ones are gonna go at low
gotcha.
stimulus.
They're already.
But when you get a really high stimulus, they all get stimulated. That's why you contract with full force. So it's both or all of them. All the fibers contract. At
Okay.
very intense
Got it.
stimulus. Okay, good. Anybody else? 10B. Okay.
29, actually.
29. Go for it.
If frequency is so fast, no relaxation occurs, a smooth, sustained contraction results called.
Complete fused tetanus or just tetanus. Either one of those should be right. Complete fused tetanus or fused tetanus, or you're looking for the word tetanus.
Okay, got it. Thanks.
Okay. Anybody else? I kind of want to move on because I also want to have time to go over the last test with people. So what I'm going to do is ask if you have any others from chapter 10, email them to me. Please remember, if you email me your review questions, they have to have an answer, because if you just send me a whole bunch of blank questions, I'm not going to send you the answer because it shows you didn't make any effort to find an answer. You got to come up with an answer that's not a Mickey Mouse answer, that's somewhere in the ballpark, and I'll tell you if you're right, and if you're wrong, I'll give you the right answer. Okay? Hannah, did you have one last one before we move on?
No, thank you.
Okay, chapter 14. Please get out chapter 14 and let's do some review there. At 3 o'clock, we do have to switch to going over old exams because I don't want to shortchange you guys this week. Okay? 14. Somebody read me one from 14. Is that number 9, Holly? from chapter 14? Okay, I'm not hearing back from Holly. Yeah, so send that to me as an email, Holly. Yeah. Okay, chapter 14. Who would like to start us off? Either in, you're welcome, Holly. Who would like to start us off with a review question from chapter 14?
Can I do an 61?
61, you gotta speak up a little bit.
Okay. Mechanical vibrations of the ossicles are converted into fluid waves at the oval window. Would that be false?
Well, do you remember what I just talked about today in the lecture on how we hear?
Yeah.
So it's air wave. So it goes from this. So let me re-explain it, and then you can get the answer. So the first thing is waves going through the air, right? Then they hit the tympanum, and it gets moved into physical movements of the bones, and then the bones cause the liquid inside the cochlea to start sloshing back and forth. That was my lecture. So now, knowing that, what would you say is the answer?
It's false.
Read the question again.
Okay. The mechanical vibrations of the ossicles are converted into fluid waves at the oval window. So it's true.
True. Right.
Thank
That was
you.
directly from my lecture. Yeah. I know it doesn't sound like what I said, but yeah, that's just what I said. Okay. It's true. Anybody else? Chapter 14. Nobody?
Number
Hannah.
53.
53, go.
Olfaction is linked to what emotional sensitive part of the brain?
I think it's olfaction.
Oh yeah, olfaction.
Okay. That's something I didn't lecture on. That is linked to the limbic system. And the significance is the limbic system is responsible for emotions and memory.
Okay.
And so our sense of smell is connected to that. So it makes things that we've smelled very memorable,
Okay.
like food or waste or death, right? Okay, so limbic system is the answer there. Anybody else? Chapter 14.
69.
Okay.
What are the two types of deafness?
Okay. The first is called conduction deafness. That's where there's a problem either with your tympanum or the bones or the cochlea itself. That's conduction deafness. The other one is called sensorineural. That is either the hair cells in the cochlea or the nerves running through to the brain or the brain itself. Anything having to do with nerve cells or hair cells would be called sensorineural. So it's conduction deafness and sensorineural deafness are the two types.
What did you say causes the conduction?
Okay, so the conduction deafness could be several. Usually it's one of two things, either a damaged tympanum, eardrum, or problems with the bones. Now, as we age, the bones go through what is called otosclerosis, which means that the hinges between the bones, instead of moving freely, move less freely. Like my mom, I have to yell at her now to have her hear me because the bone joints don't move as freely as they used to. And so it takes more force. Yeah.
Can you get
Okay.
arthritis in between those bones?
I'm sorry, say it again.
Can you get arthritis from in between those bones?
I don't think arthritis affects the ear in middle ear bones, but it's possible. Normally arthritis affects the joints of the appendages, but it could. Goodness, I've never heard of arthritis in the ossicles before, but I'm sure it's possible. I don't know. Okay. I'm going to go ahead and make the command decision. Let's jump ahead to chapter 15. We're going to finish up and then we'll start going through old exams with people who want to stick around so I can get a full lecture and review in today. So chapter 15, please get it out. Remember, if there's more chapter 14 you want to ask, send it to me as an email. Do it as early as possible and make sure you have some answer in there because if it's blank, I do not respond. Chapter 15, who would like to start us off on 15? Chapter 15. Or did you guys not get a chance to work on that because you thought it was today and not going to be on this week's test?
I did work on it. I'm trying to find it. It's in my pile of chapters.
Can we do number one?
Number one,
And
go
then-
for it, Holly.
In a neural reflex, where does the stimulus originate from?
It's usually outside the body or from the stimulus originates. That's not a great question. It's normally outside the body for most reflexes, and for visceral reflexes, it would be inside the body. But yeah, that's not a great question. I don't think it's on the test, but because it could either be the sensory receptor that I'm talking about, or where's the stimulus from actually originating from. So if it's on the skin, it would be from the.
You froze up there.
So not a great question. Sorry.
Okay.
Anybody else? Chapter 15.
Number 2,
2, read it.
where are most sensory receptors?
Okay. The answer is in your skin. Your skin has most of the sensory receptors in your body because you're picking up things that are happening to you from the outside. There are some on the inside, but mostly they're in your skin. Okay. In the dermis and epidermis. Right. Who would like to continue with chapter 15?
Number 5,
5.
most reflexes are intended to help prevent blank or to assist with blank.
Okay, what are you trying to prevent with a reflex?
Pain?
Injury.
Injury.
Injury. Prevent an injury, and pain would be associated with the injury. Or 2, what was the second half of the question?
Assist with.
Assist with movement. Assist with movement or posture. So it's either to prevent injury / pain or to help you with movement / posture. Okay, that's all the things that could be right answers there. John asked, number 17, joint receptors help to sense what 2 things? Remember, it was the angle of the joint and the tension on the muscle. So angle of the joint and tension in the muscle. Those are the. Okay, anybody else chapter 15? You're welcome, John. Anybody else chapter 15?
Yeah, number 18.
18.
In a joint like the elbow, all of the nerves, receptors, and muscles are called one.
Do you remember my lecture today? It was called myotactic unit.
I missed the first hour of the lecture because Edison is working on my street and everyone's power was off.
Oh, goodness. Well, you could have used your phone. You can still tune into this stuff on your phone. Do you guys know how to do that? You just go onto Canvas using your phone. You should still be able to click the link, and as long as you have Zoom installed as an app, you can still watch the lecture on your phone.
Okay, I can go back and watch it. That's fine.
Yeah, I'm gonna post the recording is fairly sometime today of the lecture, so you should be able to get it, even though you can't ask questions today. Let me make sure I have a reminder. Here, I just got to add a reminder on my phone to check recording. And give me just a second, folks, every week. Okay, so now I have a little reminder to go in and make sure that I get the lecture recording posted into the module. I just have to do that before I forget.
What was the number of that last question? Sorry.
What was the number on the last question?
That was number 18. Sorry.
18.
Thank you.
Okay, who would like to give our last couple of questions from chapter 15?
Number 22.
22, read it.
Where are most pain reflexes integrated?
Do you remember what I talked about? Where most reflexes are integrated, pain or other reflexes? What has more senses, your body or your head? Where's there more different things that could happen to you?
Your body.
Your body. So if it's in your body, where would it be integrated? From the neck down?
In your spinal cord?
Spinal cord, right. Spinal cord is this integration site for most pain reflexes. The only pain reflexes that are integrated in the brain are those
Up in your head.
Anybody else? Chapter 15. We have 2 more questions, and then we got to switch to going over the exam from last week.
Can we do number 30, please?
30. Go for it.
What 2 controls affect the reflexes of the physical organs?
I don't understand the question.
What 2 controls affect the reflexes of the visceral organs?
Anybody get an answer for that? It's not jumping into my head.
I couldn't find an answer for that.
I don't think that one's on the test.
I
don't
put
think it's-
automatic and somatic, but I wasn't sure if that was correct.
I don't know, but I'm pretty sure that one's not on the test because it sounds vague and unclear. I try to take vague and unclear questions off my test. So look, Catherine, if that one is on the test and you miss it, remind me next week and I'll give you a point. How about that?
Thank you.
You're welcome. One last question for 15, then we go to review of last week's test.
29.
29. Read it.
What two functions do reflexes have in the GI tract in the CV system?
Okay, so the first is to move products through the GI tract, and the second is to regulate blood flow through the blood vessels.
Thank
They
you.
act as sphincters. Okay. Hey guys, it's 3 o'clock now. I gotta cut this part off. And so this next section is going to be for anybody who wants to stick around and go over their exam from last week. If you're not interested in sticking around and going over last week's test, I'll see you later. I hope to see you guys next Tuesday. If you do want to, hey, you're welcome, John. Have a good one. Good luck with this week's test, everybody. If you don't want to go over last week's test, sayonara, I'll see you next week. And so if you are still here, what I'm going to do is I'm going to put everybody but one person in the waiting room and then hang out there and wait until I bring you back in so that you and I can go over the test. Okay. Can I assume that everybody who's still here wants to go over their test from last week?
Yes, please.
If
Yes.
you do not, time to go, time to head out if you're not interested in doing that. Okay. So we have Catherine, Erica, Jocelyn, Elizabeth. No, we lost a person. Catherine, Erica, Elizabeth, Holly, Jocelyn, Dakota. You guys all want to go over last week's test, right?
Yes, please.
Yes.
Okay. So what I'm going to do is I want to say, well, it doesn't matter. I'm just going to go who's in the upper left. Catherine, you're going to go first. What I'm going to do is Dakota, Jocelyn, Elizabeth, Erica, and Holly. I'm going to put you in the waiting room. Hang tight, and I'll be back with you momentarily. Okay?
Thank you.
You're welcome. Let's see. How do I? Let's see. Put in a waiting room. Okay. And then Erica, waiting room, and Jocelyn, put in waiting room, and Elizabeth, put in waiting room, and Holly. Okay, Catherine, so what I'm going to do is I'm going to go to your test from last week, and we'll go over the ones that you missed and the ones you have concern about. How's that sound?
Sounds great. Thank you.
Okay, so that was week 3. And that was exam 2. And oh, wait a minute. I need grade book. Great.
And it really is only 2 of the questions in particular that I can think of that I got wrong that I wanted to double check
Okay.
on. Because I
Well,
feel
let
like
me-
it
let me
was ones
find your
that we had
test.
covered.
Let me find your test first. Exam 2. What's your last name?
Sorry, Earrry.
Pari?
Yes.
Okay, my computer's really slow right now. But I also have 107 students, which is the most I've ever had. Wait, I don't see anyone with PAR in the class.
You may have me listed as Finwal Pari,
F.
FIN.
Let me go to F. Yeah, there we go, Catherine. Finwal. Try to
Thank you.
remember your Finwal Perry for me. Okay, and then boom, and then share screen, and then exam 2, and boom. Okay, this is your test, right?
Yes.
It is. Okay, tell me the question that you were concerned about first.
So, question 11.
11, too much, too fast. Okay, what do most hormones bind to? Cell surface receptors was correct.
Okay, for some reason I saw in the packet, I was thinking protein receptors.
Yes, the same thing. So in that circumstance, you should say cell surface receptors. He did talk about that. Even though I wrote down protein receptors, is it possible that cell surface receptors is the same thing? That's a tough one. But yeah, it
Okay.
was the same. And then your next one.
The next one is number 55.
55.
And that was one that we had gone over, and I believe it was number 25 in our packet.
Okay. Here's another one of those ones where I warned you guys about this. If this choice, look, see what was correct?
Two of these are correct.
Two of these
Okay.
answers are correct. You would have to say none of these other ones could also be considered a right answer. Because if you see this choice, there has to be a really good reason not to pick it. And that is only one is right and all the rest are completely and totally wrong. And I did talk about depolarization is the beginning of an action potential.
Yes.
So when you see this option, you have to say to yourself, is there a fairly good chance that there's another one that's right here? Not what did I write down in my notes? That's not what you're saying. You're saying, is there another one there that I could see as being a right answer? And if there is one, depolarization, I should pick two of these are right.
Well, I feel like when we had gone over it last week, you called it hyperpolarization, not depolarization.
No, hyperpolarization is what happens at the end of the action potential. That's when
Okay,
all of the potassium has come in.
got it. Okay.
So depolarization is the beginning of the action potential. And the key is when you see this choice, only reject it if you know for certain that all the rest of them are completely and totally wrong. But this one, depolarization, yeah, that's kind of part of an action potential. That's why two of these are correct. But
Got it.
here's what I'm going to do for you. Because you took the time to do this, I'm going to go ahead and add a point to your score.
Great.
Now you have 78 instead of 77. And that's my way of saying, good job for coming in.
Thank you very much. I
You're
do
welcome.
appreciate that.
Well, Catherine, I hope you have a great rest of your week. Good luck with the test this week, and I hope to see you back on Tuesday.
Sounds good. Thank you so much. Have a
Bye,
good one.
Catherine.
Bye-bye.